Evolution is one of the most important concepts in biology, and it's supported by multiple independent lines of scientific evidence. Today, we'll explore 4 major types of evidence that demonstrate how life on Earth has changed over time: the fossil record, comparative anatomy, embryology, and molecular biology. Each of these provides a different window into understanding how species are related and how they've changed through millions of years of Earth's history. Scientists rely on 4 main categories of evidence to support evolutionary theory. First, the fossil record provides preserved remains showing how organisms have changed over time. Second, comparative anatomy reveals structural similarities across different species. Third, embryology examines developmental patterns in early life stages. And fourth, molecular biology compares DNA and proteins between organisms. Together, these independent lines of evidence all point to the same conclusion. Life on Earth shares common ancestry and has diversified over billions of years. Fossils are preserved remains or traces of organisms from the past, giving us direct physical evidence of ancient life forms. These fossils are typically found in sedimentary rock layers, which form in a chronological sequence over millions of years. By studying fossils in different rock layers, scientists can observe a clear chronological sequence of life on Earth. The fossil record reveals extinct species that no longer exist today, and importantly, it shows transitional forms, organisms that share characteristics of 2 different groups, demonstrating evolutionary change over time. Transitional fossils are particularly important because they show intermediate stages of evolution. One famous example is Archaeopteryx, which lived about 150 million years ago. It had features of both reptiles and birds, including teeth and claws like dinosaurs, but also feathers like modern birds. This demonstrates the evolutionary transition from dinosaurs to birds. Another remarkable example is Tiktaalik, a fossil discovered from about 375 million years ago. It was a fish with limb-like fins that could support its weight, bridging the gap between fully aquatic fish and the first land-dwelling vertebrates. Comparative anatomy involves studying the similarities and differences in the body structures of different organisms. This reveals important evolutionary relationships. When we examine the anatomical features of various species, we find 2 particularly important types of structures. Homologous structures are body parts that have similar internal structure but perform different functions in different species. These structures were inherited from a common ancestor. Vestigial organs are another key piece of evidence. These are remnants of structures that served important functions in ancestral species, but are now reduced or no longer functional in modern descendants. The vertebrate forelimb is one of the best examples of homologous structures. If you compare a human arm, a bat wing, a whale flipper, and a horse leg, you'll notice something remarkable. They all contain the same basic bones arranged in the same pattern, just modified for different purposes. This shared bone structure didn't arise independently, it was inherited from a common ancestor. This pattern is called the pentadactyl limb, meaning 5 digits. This 5-fingered pattern appears across all tetrapods, or 4-limbed vertebrates, because they all descended from a common amphibian ancestor that lived over 360 million years ago. Embryology is the study of how organisms develop from fertilized eggs to fully formed individuals. When scientists examine the early developmental stages of different vertebrate species, they discover striking similarities that provide strong evidence for evolution. Even though adult vertebrates can look very different from each other, think of a fish, a chicken, and a human, their embryos show remarkably similar features during early development. All vertebrate embryos display structures like gill slits, tail formations, and similar body plans in their early stages, even in species where these features don't appear in adults. These shared developmental patterns point to common evolutionary ancestry. Let's look at specific examples of embryological similarities. All vertebrate embryos develop structures called pharyngeal pouches early in their development. In fish, these develop into gills for breathing underwater. But in mammals like humans, these same structures develop into completely different organs, including parts of the inner ear and the thymus gland. Another fascinating example is the postanal tail. Human embryos actually have tails during early development, complete with several vertebrae. As development continues, this tail regresses and becomes the coccyx, or tailbone. These temporary structures during development are evolutionary remnants of our shared ancestry with other vertebrates. Molecular biology provides the most precise and detailed evidence for evolution by examining the genetic material that all living things share. DNA and protein comparisons allow scientists to measure exactly how related different species are at the molecular level. 3 key observations support evolution. First, all living organisms on Earth use the exact same genetic code, suggesting all life descended from a common ancestor. Second, the degree of DNA sequence similarity between species reflects how closely related they are evolutionarily. Species that diverged more recently share more similar DNA. Third, scientists can use molecular clocks, which measure the rate of genetic mutations over time, to estimate when different species diverged from common ancestors. DNA comparisons provide quantitative evidence for evolutionary relationships. One of the most striking examples is the comparison between humans and chimpanzees. These two species share approximately 98.8% DNA similarity, which confirms they share a close evolutionary relationship and descended from a recent common ancestor about 6 to 7 million years ago. Another powerful example comes from studying a protein called cytochrome C, which is found in virtually all organisms that use oxygen. This protein performs the same essential function across all aerobic life, but the exact sequence of amino acids varies slightly between species. The number of differences in cytochrome C sequences correlates directly with evolutionary distance. Closely related species have nearly identical sequences, while distantly related species show more differences.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.